All right, everyone. Welcome back to the second part of this LBI meeting. And it's my great pleasure to introduce our next speaker, Lynette Lim. So Lynette joined VIB-KU Leuven as a group leader in January 2020, following a postdoc career in the Institute of Neuroscience in Alicante and King's College London. And she describes herself as a neurobiologist who likes to solve puzzling questions. And indeed, her research group tries to unravel the metabolic and transcriptomic programs that shape neuronal diversity and circuit assembly in the developing mammalian cortex. There too, Lynette employs an interdisciplinary approach that combines mouse genetics, virus tracing, cell-fate mapping, metabolomics, and single-cell RNA sequencing with cutting-edge imaging techniques and bioinformatics to define how developmental programs influence cortical circuit formation. Lynette, we are looking forward to your talk. Please go ahead.
All right. Thank you very much. Let me just make sure that I can properly share the screen. I hope everyone sees this screen at the moment. Thank you very much for the organizers for this meeting invite. Now, let me see. Okay, we are interested to understand how brain circuits are built. The part of the brain that we work on is the mammalian neocortex, which houses some of the most complex circuitry. The cell type that my lab study extensively are inhibitory neurons. So unlike the majority of the neurons, which are excitatory and projects long distances to other areas of the brain or within different parts of the cortex, these inhibitor neurons are mainly locally projecting. Therefore, they are usually termed as interneurons. And one of the key functions as interneurons would be to integrate relevant information that comes into our brain. So in our lab, we like to use the analogy of traffic lights to think about this problem. If you consider there are obviously a lot more roads in a city than traffic lights. But however, when you have one traffic light that goes a bit malfunctioning, you have a huge traffic disruption. So if we were to extend the analogy of traffic lights a bit further, we can ask the question, how many types of neurons really exist, okay? In the last 100 years, there have been a tremendous effort to understand neurodiversity in the cortex because we see this as the fundamental building block of a greater circuit. If we know all the playing partners, we can then infer how they all come together. The largest effort to document this comes from the Allen Brain Institute, who have now performed single-cell RNA sequencing of more than 1 million neurons in the cortex and hippocampus. And one of the key findings that is largely underappreciated is that lower abundant cell population have tremendous high diversity. So I give you a bit of a number analogy here. In this 1 million neuron data set, there are 160,000 inhibitory neurons with 118 types. In contrast, even though they have sequenced over 900,000 excitatory neurons, there are only about 235 types. So the same number of cells, inhibitory neuron diversity normalized to this would be 5 times more. Okay, so if now we zoom into these inhibitory neurons, again, we find what I've just told you. The most abundant class usually are not the ones with the highest diversity. We know, for example, the parvalbumin expressing biochemical class are the most abundant among all inhibitory neurons in the cortex, yet they exist to have 5 what the Allen Brain Institute call MET type. And MET is a description that links transcriptomics, morphology, and electrophysiology, therefore MET. So the most diverse cell types appear to be those expressing the neuropeptide somatostatin. And we'll come back to this a little bit later on somatostatin interneurons. So as I mentioned to you, our lab, we are interested in how this diversity is generated. And let me give you now a few models on how we think about this problem based upon current data. The first model I would like to put forth to you is that the identity of different subclass of neurons are already present shortly after neurogenesis, so at post-mitotic division. And it is mainly the progenitor itself that change over time to generate different types. And each type would then unfold a conserved program over time. There's some evidence to suggest this is true for excitatory projection neurons because cell identity is strongly correlated to the time of birth. For inhibitory neurons, this is unlikely to be true because we know that the same progenitor can make very different subtype of neurons. Okay. In the second model, the complete specification happens at various stages. While the main class are determined shortly after a cell becomes post-mitotic, each subclass acquires its identity depending on a long post-mitotic program that would be cell type specific. Under such a framework, we would expect to find a set of shared as well as unique developmental program for different cell types that are within the same biochemical class. So at least for inhibitory neuron, we are interested in understanding what are the shared and unique developmental trajectory for each cell type, importantly, post-mitotically, okay? So we know a lot about the development of inhibitory neurons. This is a bit of a general program that summarized here over over the work of many labs over many years. Okay, we know that shortly after being post-mitotic, first we know that they are born not in the cortex; they are born outside the cortex. Shortly after being post-mitotic, each neuron travel a long distance to get to the cortex, where they would find their laminar location in the cortex, following a transition from tangential migration to laminar allocation. Sometime within the first postnatal week, there's a wave of programmed cell death, and eventually followed by axonal expansion and circuit refinement. What I just described to you is a maturation program from a population perspective, in that every single inhibitory neuron would do this, also known as a generic program. However, we know that this is not, there are unique features in different neurons. For example, in 2018, we've published that the tangential migration route preference of different cell types can be seen as early embryonic stages around E16, okay? Because of this, we became very interested to ask, other than migration route pattern, what are the other unique post-mitotic developmental programs that is specific to different types of neuron? And even though what I show you here is a generic process that all cell would go through, what is the tempo or the timing of each step? Is it the same for every cell type, or could this be dramatically different? Now, with the advancements of single-cell technology, we can now start to tackle these important questions that we could not before address. And we use somatostatin inhibitor neuron as a model to study this. As I alluded to you, they are highly diverse. So the experimental paradigm is that we dissected out the developing cortex at E16, P1, and P5. We enrich particularly for somatostatin neuron, because as I said to you previously, if we just look at all the cell types, we're unlikely to catch them because they are minority population, even though they are highly diverse. So we purify and enrich for these cell types at 3 developmental stages and perform single cell RNA sequencing. In our collection, we have about 9,000 of these enriched cells, and we're able to discern easily 12 types of cell within this single biochemical class, even as early as E16.5, okay? When we saw this diversity in our data set, and this is very early in development, we started to ask why this has not been documented before. And let me give you one of such example. Okay, in 2018, again, using single-cell RNA sequencing, Gorfischer's lab, Christian Meyer here was the postdoc that worked on it, who now has his group at Max Planck. He basically purified interneurons using a different Cre driver line. But in his collection, he has about 500 somatostatin cells ranging from E18.5 to P10 in the mouse cortex. Now, here I show you here the original papers, TSN plot, where they describe one population called MGSST. And within there, they describe 3 populations of somatostatin cell. Okay, they don't find 12 type like we do, even though they have a timing quite similar to what we have. Okay, now we downloaded these raw data from this 500 somatostatin neurons in that data set, and we use our trained classifier to re-annotate this data set. And lo and behold, we do find all about 11 out of the 12 types, even with just 500 neurons, with an accuracy ranging from 77 to 95%, depending on the cell type we're talking about. So this shows you that when you don't sequence enough cells, right, you largely underestimate the diversity, and even in developmental stages where the diversity might be right there. Okay, similarly, what we could do is now compare the cell identity to the adult identity. So we're taking developmental identities to link it to the adult identities. And what we find is that many of these identities has corresponding matches in the adult. So this data set, what they tell us is that we can now clearly resolve diversity during development. Now, this is important because what we're asking is, if different cell types have distinct developmental trajectory. So if we can't discern identity in early stages, there's no way to answer this question. Okay, right? So now, to answer the question, do different types of neurons have distinct developmental trajectory? We perform a pseudotime analysis. Okay, this is a very busy slide, and I'll take you through this. Let's focus first on panel D and E, okay, which shows us that we can arrange in pseudotime, moving from low to high in 2 axes, okay, in diffusion component 1, that is in your x-axis, and diffusion component 2, that's in your y-axis. What this shows us is that as we move from the pseudotime from very low to very high, it matches corresponding well with the sample going from E16 to P5, okay? The second thing that it shows us is that we have almost 2 orthogonal pseudotime development, as if cells are developing in 2 almost orthogonal and not really related stages. And if we now go back and look at who are the cells that are in the first branch, so the one going on the top, or going more to the right side, what we see is that, in fact, the long range, these cells that are annotated as LPR, which are the long range projecting neurons, versus those that are either annotated as Martinotti and non-Martinotti. Okay, to illustrate this point a bit further, what I'm showing you here is a heat map on gene expression for each branch, arranging again by pseudotime from left to right. And if we look at so let's just focus on the long range branch. We see that you have some genes that are turned on. I don't know if you can see this, but NPY, for example, is turned on rather late in this pseudotime. And you have some genes like LYPD that is turned on quite early in the pseudotime, but this is quite unique to this long range branch, okay? And so now I talk a lot about this branch that appears to have its own developmental trajectory, this LRP branch. What exactly are these? And why do these inhibitory neurons look so different than others in this single cell plot? As alluded to in the introduction, I told you that inhibitory neurons are locally projecting, they are interneuron. However, this is not completely true. We know that there's a very small population that in fact long range, okay, and they can project to other cortical areas. here, what I show you is an experiment that we've done that others have also done in a slightly different way. So we inject anterograde tracer, a virus, to infect the motor cortex. And this virus expresses T tomato in a Cre-dependent manner using only a promoter that is specific to GABAergic neurons. As you can see, we inject very specifically in the motor cortex, and we look at the axonal projection outside the cortical area, and you could see that there's a lot of projections in the striatum, as well as we know that these neurons are also project into different cortical areas. Clearly, one project in our lab now is to look at candidate genes that appear to be exclusively expressed in these long-range cells, and to understand why the development is so different and what guides their development. So let me come back, to the slide, to this slide that I've shown you before, as a way to highlight key developmental milestone of inhibitory neurons. As I mentioned to you, this is a generic program. Given that now we can describe both shared and unique gene expression pattern over development, we can ask for each of these key milestone, one of them being laminar allocation, are there different timings for different subtype? And to put it in another way, do all the cell type arrive at their laminar location at the same time? Ideally, to do this experiment, what we would need to do is to be able to mark every single cell type, track them live as they move. But obviously, this is not a feasible experiment. And so luckily for us, we have some data that help us. So I go back to this slide that I've shown you previously to look at the diversity of adult somatostatin inhibitory neuron done by the Allen Brain Institute. What they have shown us is that contrary to a long-standing dogma that inhibitory neuron has no laminar distribution preference, these data sets actually show us that somatostatin subtypes have clear laminar preference. As you can see, some of these are very discrete, only in deep layer. So if this is the case, now we can ask how early developmentally is this pattern of laminar allocation established? Okay, let me remind you again of the question we're testing. If the developmental trajectories are molecularly different, does this mean that the completion of certain milestone using laminar allocation as one would also be different? To answer this question, we collaborated with Resolve Bioscience, who is a spatial transcriptomic company, by using their molecular cartography method. Essentially, this is similar to a single molecule fish, where up to 100 different transcripts could be barcoded using combinatorial fluorescent probes. And through sequential imaging, coloring, and removing the color, this will allow us to extract the transcript expression in a tissue setting. So using this method, we were able to image rather large region of interest, which give us close to 40,000 cells in the developing cortex at P5. However, because only a subset of this are somatostatin, in this approach, we end up with about 2,500 cells that we could annotate and match back from spatial transcriptomic data to a single cell sequencing data set. So to compare this to the adult data set, what we did was to perform what we call a chain link method, first matching our spatial identity to our RNA-seq identity using 10x that we use for single cell sequencing, not spatial, and then linking this back to the adult data set that was previously published by the Allen Brain Institute. And so you could see with this kind of chain mapping, we could link up spatial identity we have at P5 to spatial identity we have in the adult. So what does it look like? So here in panel D, I show you all the cell identities we've uncovered with our spatial data set. Okay, considering the number and the gene cell number and the gene number examined, this is much less than using kind of an unbiased single cell sequencing. We do not always have a clear one-to-one matching, but for some subclasses, this matches up well. For example, you can easily see, hopefully, but this is a bit small, you can easily see that for these long-range group that I've talked to you about, they match very well to what we annotate as the somatostatin chola one. Similarly, for a group like the non-Martinot T5 here annotated, this mostly categorized to a somatostatin ERBB4 group in our spatial data. So now we can get to answering our question. When we look at laminar distribution at P5, comparing it to the cell type counterpart in the adult, what we can see is that for some cell types, the laminar allocation is very similar at P5 versus the adult. In contrast, others are highly different. For example, these somatostatin ERBB4 type 2 that almost do not have the matching to the adult. So in conclusion, what I have shown you today is that inhibitory neurons are of the same biochemical class, has cell type specific maturation program post-mitotically. And this is not just a description at the molecular gene level, but we can also link it to the timing of acquiring key milestones such as laminal allocation. With that, I'm out of time, and I'd like to really thank our funders, the lab members that did the work, our collaborators both in academia as well as outside, such as Resolve Bioscience. And I'm happy to take any question. And one last thing, we are recruiting, so feel free to contact me.
Thank you very much, Lynette, for this wonderful talk. Unfortunately, we don't have much time for questions. Maybe you can address one. In the chat box, someone is asking, are all these different subtypes of inhibitory neurons also different on their morphology and or firing patterns?
Yes, absolutely. That was one of the key aspects of the Allen brain data that shows this MET, which means this morphology and electrophysiological properties.
All right. Thank you very much.
Yeah.
I think we can immediately go on to the next speaker then. And that is Wouter Peelaerts. I hope Wouter is ready. Yes, so Wouter is a senior postdoctoral researcher in the research group for neurobiology and gene therapy in the Faculty of Medicines. And during his PhD, he gathered seminal evidence for the link between abnormal alpha-synuclein species and Parkinson's disease and multiple systems atrophy. And after a 3-year stay in the Van Andel Institute in Michigan, he recently returned to KU Leuven to set up a research program focused on the identification of infectious disease triggers, pathogenesis of multiple systems atrophy, and preclinical drug testing for both Parkinson's disease and multiple system atrophy. So I think today he will introduce us into his work on how peripheral infections may trigger multiple system atrophy. Thank you very much, Wouter, for presenting here today. We are looking forward to your talk.
Okay, thank you. Thank you, Lise. Thank you for the introduction. I'm just trying to share my screen here real quick. All right, there you go. Just let me know in case these slides are not showing. And so I think I've heard a lot of interesting talks here today and really exciting work on a wide range of topics. And I think I'll be shifting gears here just a little bit. As I just mentioned, Lise, I'll just be discussing how infections can trigger synucleinopathy, and more specifically how UTIs or urinary tract infections could act as a trigger of MSA. So I'm a postdoc in the lab of Vele Bakeland here at KU Leuven, and so a lot of this work is actually also from my previous postdoc at the Van Andel Institute. And so we're still doing a lot of research here on this topic, and we're generating new and a lot of exciting data in the context on how infections can trigger synucleinopathy in general. But here I'll be focusing on how UTIs can be involved in MSA. So I don't think MSA is a very well-known disease, and I think that's mostly because it's a relatively rare disease. It occurs in about 1 in 25,000 people. It's a neurodegenerative disease. It's also a demyelinating disease that affects both oligodendrocytes and neurons in the central nervous system. And there's currently no therapy. There's even no effective symptomatic treatment just to alleviate some of the many symptoms that people with MSA have. The average age of onset, or the point where the disease is usually diagnosed, is around late 50s, early 60s. We don't really know how MSA is caused. There are no known genetic risk factors or environmental risk factors, and even there are also no good biomarkers to make an accurate or good diagnosis of multiple system atrophy. So what is very well established in the context of MSA is the neuropathology. And so if you look in the brain of people with MSA, you'll find widespread deposits of alpha-synuclein inclusions, synuclein aggregates, in the, for instance, here, what is shown in the brain putamen of a patient with MSA. So this, for instance, this slide here was stained for alpha-synuclein in a control subject, which is, as you can see, virtually negative. Then here in the brain putamen, you can see the white matter very densely stained for synuclein protein, which then you will find within these oligodendroglial cells, which is a very typical feature of multiple system atrophy. So because the involvement of synuclein, MSA is a synucleinopathy, and there are also other types of synucleinopathy next to MSA, which are Lewy body dementia and Parkinson's disease. And so the biggest difference here is that in case of LBD and PD, synuclein deposits within neurons, making these primary neuropathies, whereas in the case of MSA, synuclein again is depositing within these oligodendrocytes. And so we've learned a great deal from these diseases, from these synucleinopathies, so MSA, LBD, and PD, because they have multiple shared disease mechanisms. And so one of the, let's say, most interesting features, or I should say the most studied features, is the aggregation of synuclein protein and how this affects cellular health and pathology, and how this eventually then results in disease. And so in short, so synuclein is basically present as a soluble monomer that then starts aggregating because of unknown reasons into high molecular weight assemblies that then can form stable seeds to amplify and propagate these longer inclusions, which then will eventually deposit into these very rich, dense inclusions that are then detectable in brain tissue, as I just showed in the previous slide. One important mechanism that I would just like to mention here, because I'll be discussing this also later in one of the slides, is the cell-to-cell transmission, or the transmission hypothesis of synucleinopathy. So basically what that means is that in response to an unknown trigger, synuclein starts to accumulate and aggregate intracellularly in a cell, and then this leads to the accumulation and the aggregation of the protein, and to leading to some sort of vicious cycle where the protein basically starts to replicate and even spread from one cell to the next. So whenever these inclusion starts amplifying, you will get these longer fibrils that will break in shorter fragments, and then these will then spread from cell to cell, again leading to pathology in a spatio-temporal manner. And so this is known as the transmission hypothesis. Some people also refer to this as the prion hypothesis or prion-like hypothesis, which is also very well known for other types of proteinopathies. So multiple system atrophy is also characterized by a great clinical variety. And so when people come to the clinic, they usually present with two different clinical subtypes, which can either be the cerebellar subtype of multiple system atrophy, and on the other hand, you'll also have the Parkinsonian subtype of MSA. So in that first case, there is olivopontocerebellar atrophy, which then corresponds to a high burden of synuclein pathology within the cerebellum and the brainstem here. And here, on the other hand, in case of the Parkinsonian subtype, there's a high burden of synuclein pathology in the putamen. So again, corresponding with the clinical picture and the neuropathology. So as I mentioned, there is some heterogeneity in multiple system atrophy. And so when people again come to a clinic, they usually present one of these very many features that can present during the disease course. And so diagnosis of MSA is usually made when the disease is already significantly progressed. So when these Parkinsonian or cerebellar features present, then the diagnosis of MSA is usually made. But the thing that I just want to point out here and mention is that this long premotor prodromal phase here of multiple system atrophy is that during this phase, people usually present with peripheral autonomous features. So not necessarily these central neurological features. And so some of these features that really stand out are urogenital symptoms, respiratory symptoms, and gut-related symptoms. And for instance, patients with MSA have urinary retention, men have erectile dysfunction, and women have anorgasmia. And this is very typical of MSA, and you won't, for instance, see in a person with Parkinson's disease. What's also really striking here is that there is a strong involvement of infections, and even here during the prodrome, people will have very frequent and chronic UTIs. So the fact that we see, this peripheral involvement early in the disease course actually makes us believe that the disease could potentially start within these sites. So I just want to mention here that we think that it is very important in terms of studying the etiology, just going back to the sites and potential triggers from where we believe the disease might start, and then subsequently spread via connected innervation to the central nervous system. And also here in that context, we know that infections are prominent in MSA, also here within the periphery, in terms of in the context of UTIs, again, during the prodrome, here the premotor phase of multiple system atrophy, but also during the disease where people have recurrent UTIs and chronic UTIs. And in fact, more than half of all people with multiple system atrophy have recurrent UTIs, which is unusually high. And then even here at the end of the disease, one of the two leading causes of death is in fact urosepsis. And then more recently, there was this study from a Chinese group that very interestingly found that there is synucleinopathy, so synuclein pathology within the urinary bladder. And so you can see here a staining that was done for the same marker that I showed initially, so phosphorylated alpha-synuclein in the urinary bladder tissue. And they showed that this was very specific for people with multiple system atrophy. They could not find this type of pathology for people with Parkinson's disease or progressive supranuclear palsy, or in any of the healthy controlled individuals that they examined. So putting all of this together, we started wondering whether MSA, for which we actually don't know what causes MSA, whether this disease could be triggered somewhere in the periphery, potentially by an infectious trigger, and potentially by a urinary tract infection. And so therefore we started modeling this in vivo. We started infecting wild type animals by inserting catheter and injecting and infecting these mice with uropathogenic E. coli, shown here. And then over time, over 24 hours, you can measure the infectivity of these animals. And here you can see that over these initial 24 hours during the acute infectious phase, that these mice develop a relatively strong infection. And then so correspondingly, when we look at synuclein expression in the urinary bladder of these mice, we can see also that during these acute infections, synuclein, or the expression of the protein alpha-synuclein, so this potentially pathogenic protein, is suddenly increased and is acutely expressed within the infected site. And so this just shows here the Western blot for animals injected with PBS, and then animals infected with uropathogenic E. coli. You can see that the total levels of alpha-synuclein here go up in response to infection. And even more interestingly here is that when we isolate the aggregated alpha-synuclein from the urinary bladder, we see that during infection there is aggregated alpha-synuclein in response to the infection. So we were very excited about this result, but also quite puzzled because this was the first time that, to our knowledge, we found a direct link between a potential pathogenic protein and something actually rather stochastic, an infection that would come in through the environment and then have its influence or its effect on the periphery. And so we then started looking where the synuclein was coming from. And so initially we started looking at neurons because we know that this is where synuclein is endogenously expressed, but we couldn't find anything, we couldn't find any increase or no decrease. And in fact, if there was anything, then the expression was relatively stable within neurons. So then if we look more closely to the infected tissue, we then found these polynuclear granulocytes, and in fact, these are neutrophils. So these stand for myeloperoxidase, which is a marker for neutrophils. And we found that these innate immune cells were expressing alpha-synuclein. And so it's actually the innate immune system that was bringing in alpha-synuclein. And this is just a different method or a different way of analyzing this, where we show that here in the mucosal layer, here in the infected tissue especially, that synuclein, again, expression goes up, leading to a higher presence of alpha-synuclein, again, this pathogenic protein within the urinary bladder. So, sorry. So neutrophils or granulocytes in general, they have multiple mechanisms by which they can fight off against invading pathogens. And one of those mechanisms is to form neutrophil extracellular traps. And so this is basically a mechanism by which they release their DNA, they decondensate their DNA, and then also release lytic enzymes, reactive oxygen species. And by doing so, they trap and capture and kill the bacteria that are causing the infection. And so here in this image, you can see two of these nets, two of these neutrophil extracellular traps. And we also stained the tissue here for alpha-synuclein. So all of these green cells are neutrophils. H2B is an histone marker showing that the DNA here is being released in these two cloud-like assemblies. And then alpha-synuclein, which we actually found also within these nets. This is just the fluorescent intensity profile showing the overlap of these three markers. And this here is a higher magnification of this little part here of this net. Again, showing that alpha-synuclein is being released from these cells and is being deposited within the extracellular space or within the infected tissue. So after seeing this, we'd also started modeling this in a cellular model. So we just then used granulocytes that we differentiated in vitro, and we attached a fluorescent tag to alpha-synuclein, and then we did live imaging to see if synuclein was being released. So here you can see a bright field image of fluorescent alpha-synuclein. You can see the total alpha-synuclein, and here you can see the protein, the alpha-synuclein that is being released. So over time, upon infection, so these are infected cells, you can see that synuclein, this protein, again, is being released from these cells. So this was all done in wild-type animals and in cell culture models. And so then the next question obviously was, whether this was also happening in human cells and in human urinary bladder. And so the short answer to the question is yes. So actually we did find, again, also here, expression of human alpha-synuclein within human neutrophils. So if they're stimulated with PBS, if there's no trigger, you can just see endogenous expression of this protein. But then if you stimulate them with your pathogenic E. coli, with UPEC, you can see again that these cells start basically spitting out their DNA, and that at the outside of this webs, you can again find alpha-synuclein. Here then at the right side here of this slide, you can see images from actual cross sections of human urinary bladder. The top is just a healthy control with circulating resting neutrophils, in which it is actually very hard to detect the endogenous alpha-synuclein. But then in a case here with cystitis, you can again see these cloud-like myeloperoxidase positive cells. And then here at the outside, again, you can see synuclein being deposited. So in short, just as a summary for this part, we found that infections can actually trigger the expression and the release of a protein that is associated with a very large group of diseases. And even more so, we also found that this protein can actually aggregate within the infected site. So this then, of course, made us wonder whether it's possible that the aggregated protein that is found within the infected site can then spread or escape the infected tissue and then spread to the central nervous system as part of this cell-to-cell transmission hypothesis of synucleinopathies. So in order to do this, we then started modeling, or we then created a new animal model to study the spread or the propagation of alpha-synuclein from the periphery to the central nervous system. And so we started by using these humanized alpha-synuclein transgenic mice. So these mice do not express any endogenous rodent alpha-synuclein, but they only express the human form of alpha-synuclein at normal expression levels. And in addition to this here, and secondly, we also created a transgenic model that we think would be more susceptible towards developing MSA by treating these animals and injecting them with viral vectors that are specific for oligodendrocytes. And so by targeting oligodendrocytes in the central nervous system, we then would express a bit more human alpha-synuclein to generate a model that might be more susceptible towards multiple system atrophy. So practically what that means is that here in this, what we call the human alpha-synuclein model, these are humanized and synuclein transgenic mice. There is just a normal expression here in the gray matter. So this is neuronal alpha-synuclein for human alpha-synuclein. And then in the white matter, we can also find some moderate expression, some low expression of alpha-synuclein here in these white matter oligodendrocytes as shown by the GSD pi marker and human alpha-synuclein. Then the second model, which we then call the ODC alpha-syn model for oligodendrocytes, we have simply more expression of human alpha-synuclein here in these white matter columns, as again shown here by this slide. So again, two models that we then used to study the potential propagation of synuclein. Then secondly, what we needed is the actual aggregates. So we want to inject the aggregates into the urinary bladder and then look if they can spread to the central nervous system. And so for this, we took the aggregates from human MSA brain. We took them out of the brain, we isolated them and purified them, and then amplified them in vitro by an amplification mechanism called PMCA, by which we basically sonicate and fragment these large inclusions into smaller pieces, and then use these pieces as a template and seed with recombinant monomers. And this cycle is then repeated many, many times to then eventually generate synuclein fibrils that are derived from a human patient brain. And so then we injected these fibrils into the urinary bladder of our mouse models, and then just waited for 3 months, 6 months. And then eventually after 9 months, we noticed that these animals were developing multiple behavioral deficits. So firstly here, we noticed that these animals were developing urinary bladder deficits. So there were changes in the total voiding volume for the animals here. So the ODC alpha-syn mice and then the humanized alpha-syn mice injected with either the fibrils and as a control, the alpha-synuclein monomers, just to make sure that the injection on its own was not causing any troubles. But so by injecting only the fibrils, we can induce a voiding deficit reminiscent of bladder dysfunction. But even more so, these animals started developing gait dysfunction, shown here, but also even they had dysfunctional fine motor control, again, indicative of a MSA-like phenotype. And then when we looked in the central nervous system, more specifically in spinal cord and brainstem, we also found that these animals started developing oligodendroglial inclusions, as shown here by these pathological markers. So then what we've shown is basically, first of all, we've identified a trigger by which synuclein aggregation can be initiated via an innate immune mechanism. Secondly, we then also showed that it is possible for synuclein to actually propagate and escape the periphery and transmit to the central nervous system. But what we didn't do at this point is to look at whether infections themselves can trigger multiple system atrophy. And so that's the last experiment we did. And we used the same transgenic models in the same setup as I just explained before. And I'm just going to explain here the infection paradigm. I'm not going to go into these results here. But what we did here is we infected our mice with a chronic infection paradigm. So they were initially infected the first time, 7 to 8 weeks after birth. Again, our different groups are different models. Then we let these animals just sit in their cages for 4 weeks after they received antibiotics. Then again, they were challenged the second time with another infection. And then again, they received antibiotics. And then we looked at the same time point 9 months after these infections, and then looked at whether these mice and these models were developing synucleinopathy. And so what we found is that virtually in every condition that we tested, or every model that we infected, there is synucleinopathy or an increase of pathology in any of these mice that we tested. So the normal, that's a humanized alpha-synuclein mice, but also these animals that were more susceptible towards MSA, we found that they all develop oligonucleal inclusions along the entire spinal cord, and even all the way up to the brainstem. So in conclusion, I think we can say that infections trigger de novo synuclein aggregation via an innate immune mechanism, where we also found that synuclein can be released from these innate immune cells and deposit within the infected tissue. We've also shown that synuclein aggregates can actually transmit from the urinary bladder to the central nervous system in vivo, and even more so that UTIs themselves, recurrent urinary tract infections, can trigger synucleinopathy in vivo. So I think this is really exciting work, and obviously there's a lot of people involved here with this work, especially people from Patrick Brunden's lab at the Van Allen Institute. There's really a lot of people to mention here. So Lena Brunden Laboratory, Scott Hudgren's lab, Magdalena Ivanova, and then of course here also Veerle Bakeland in the lab here at KU Leuven, and then all of the sponsors that made it possible to do this research. And with that, and if there's any time left, I am happy to take any questions.
Thank you very much, Wouter. I think that maybe we have time for 1 or 2 small questions. So in the chat, Lucas van Oudenhoven is asking, or is actually mentioning that there is similar research in the gut-brain axis, and he is asking whether you have looked at the gut too.
Yes, so me, myself, and I think many other people are very interested also at looking at gut-to-brain axis. These are also some of the experiments that are also happening here at the lab, where we're studying transmission from the gut to the central nervous system. It's a bit of early work at this point, but I'm certainly we're going to get some similar and exciting results in that regard as well. In terms of transmission itself, there have been several studies that have shown that if you, for instance, inject these fibrils in the gut, that they can transmit through the vagal nerve to the central nervous system, and then even cause Parkinsonian features in vivo. It's been debated how this happens in real clinical settings, but also there is some evidence that this can indeed happen. But this, I think it's a very relevant question, certainly in the context of Parkinson's disease as well. And I'm very curious and interested to see what's going to happen more here in this, this area for this type of research in the near future.
Okay, and then maybe one more question from Samal Bisenova. We know now of the existence of different neutrophil subtypes, some of them with an enhanced inflammatory profile. Have you tried characterizing these bladder infiltrating neutrophils?
Well, that's on our planning. That's very high on the to-do list. So this was a bit of a serendipitous finding. We weren't actually looking for innate immune cells expressing alpha-synuclein or any component of the immune system being involved in all of this. So this was actually, like I said, a sort of coincidental finding. So we've been looking at some of the mechanisms that might lead to pathology and then subsequent disease progression. And so now the next question obviously will be looking at how the immune system is involved. For instance, what type of neutrophils might be involved to characterize this in more depth. Again, here, I think this is a very relevant and important question that we'll be addressing in the near future.
All right, thank you very much for your very interesting talk and for answering these questions. Any more questions can be asked in the networking tool or via the chat box, or just contact Wouter. And I think we have to go on with the next speaker.
All right,
Yeah.
thank you.
Thank you, Wouter. Our next speaker is the amazing Boushra Dalile. She obtained her Bachelor in Behavioral Studies, Psychology from Swinburne University of Technology in Melbourne, Australia. She then completed a Master of Science in Cognitive Neuroscience at the University of Skövde in Sweden. She was awarded the Global Swede Award by the Swedish Institute, something like the Nobel Prize for Scholars, and the Best Master Thesis in Cognitive Neuroscience. After that, she was a guest researcher at the Max Planck Institute, and since 2017, she's a researcher at the TARGID, Translational Research Center for GastroIntestinal Disorders. Boushra defended her PhD 2 weeks ago, so I give you now the stage, Boushra. The first presentation as a doctor.
Indeed. Thank you very much for this nice introduction. I think everybody can see my slide. So similar to Wouter, I'll be taking a little bit of a different look on psychobiological functioning. And here it will be from the perspective of the gut and the resident bacteria, actually. So let me see. Voilà. So although food has classically been perceived as a means to provide energy and building material to the body, its ability to prevent and to protect against mental illnesses is starting to be recognized. So to give you some statistics, did you know, for instance, that the consumption of a healthy diet comprising plenty of fruit, vegetables, legumes, fish, and whole grain products was associated with the 32% decrease in the risk for depression and up to 40% decrease in the risk for cognitive impairment? Now, this is all nice, but the question is, how does this come about? It has now become indisputable that our brains and our guts are connected in a bidirectional manner, and that is thanks to multiple neuronal and hormonal and immune signaling mechanisms. But recently, the trillions of bacteria that is residing in our colon is proposed to be an important mechanism in this gut-brain connection. Now, we live in a symbiotic relationship with the bacteria in our gut as we provide them with a warm and stable environment to thrive, and in turn, they help to train our immune system to synthesize vitamins, metabolize drugs, and also to break down certain food components that we cannot break down ourselves. As an example, when we consume a diet that is rich in fruits, vegetables, legumes, and whole grain products, the dietary fiber component of such foods actually passes undigested through the gastrointestinal tract and escapes digestion in the small intestine as we lack the necessary enzymes to break it down. Instead, it goes all the way down to the colon, where it gets greeted by the gut bacteria that help to break it down for us in a process called fermentation. So the fermentation of dietary fiber in the colon by the gut bacteria predominantly results in the production of short-chain fatty acids, or SCFAs. And these are mainly acetate, propionate, and butyrate. These molecules are of crucial importance to the health of our colonic cells as they provide them with the necessary energy. And they also, for instance, butyrate helps to protect the colon from the development of colon cancer or cancerous cells. Now, most of these short-chain fatty acids are already metabolized in the colon, and the fraction that escapes oxidation travels to the portal circulation and ends up in the liver, and then a fraction of that ends up in the systemic circulation, some of which can actually make it to the brain. Now, most of what we know about this microbiota-gut-brain axis comes from animal studies. For instance, we know that the composition of the gut microbiota and its capacity to produce short-chain fatty acids influences how quickly animals learn new information and how well they memorize them, how they respond to stress, and whether they end up developing depressive or anxiety-like behavior later on. In contrast, in humans, we have primarily observational or correlational evidence, and we don't yet really know how changes in the gut bacteria influences brain function. And that, but what we do know is that they cannot themselves, under physiological conditions, make it to the brain to influence psychobiology. And that is due to the presence of physical barriers, for instance, the intestinal barrier and also the blood-brain barrier. However, we do think that short-chain fatty acids could act as messenger molecules in this microbiota-gut-brain relationship in a way that they, for example, transduce the gut microbiota signal and influence brain function. So for the past few years, I've been studying these molecules in relation to psychobiological functioning, and I was asking the main question: how does what we eat influence how we respond to current and future threats? Now, to do this, I looked at two psychobiological processes that underlie the development of a wide range of psychopathologies. I looked at the human stress response, which is a response to current threat of an uncontrollable and unpredictable nature that exceeds our current coping resources. And this results in the activation of the hypothalamic pituitary adrenal axis and the subsequent release of cortisol, which regulates our fight or flight response to the stressor that we encounter. In addition, I also looked at the development of fear and its extinction, which, similar to stress, also results in sympathetic activation and the recruitment of various brain regions to launch immediate fear responses, to regulate appropriate behavioral responses, and to provide contextual or spatial information, and also to process sensory information to facilitate future-oriented responses to threatening stimuli. Now, both of these psychobiological processes are critical to survival, but when we fail to adequately recover from stress or habituate to known stressors, or when we fail to inhibit our fear responses, when we know that the stimuli we're encountering are not threatening anymore and not predictive of an aversive outcome, some people can end up gravitating towards pathological responding and eventually develop psychopathologies. So in order to study the stress response in the lab, I asked participants to place their hand in cold water and to perform mental arithmetics for a period of about 10 minutes, all the while they were being videotaped and receiving negative feedback from the experimenter. This resulted in a robust activation of the hypothalamic pituitary adrenal axis and the subsequent release of the stress hormone cortisol, which was later detected in saliva samples. I also asked participants how they were feeling throughout the stress induction procedure, so subjective responses, and I also collected blood samples throughout the study day to measure the levels of short-chain fatty acids in the systemic circulation. Now, in relation to stress, in relation to fear, I employed a fear learning and extinction paradigm. And basically, this involves presentation of two neutral stimuli, such as here a blue and a yellow light, one of which is repetitively paired with an aversive outcome, and that is a mild electrical stimulation. After multiple pairings, with the electric stimulation, the previously neutral stimulus becomes known as the positive conditioned stimulus, and the neutral stimulus that was not paired with an electric shock is a negative conditioned stimulus. And as you could imagine, the sole presentation of the blue light later on would result in a conditioned response. So just the mere sight of the light would result in higher responses at the physiological and subjective levels, which we call conditioned responses. And this is basically what happens when we are afraid of, or models when we are afraid of a certain stimulus in our environment. Now, this fear can be extinguished, and this happens when we in the lab present these neutral stimuli without anymore the pairing with the electric shock. And this also models what patients undergo when they participate in exposure therapy, for instance. And here what we see is that over time, the conditioned response goes down. Now, what happens during fear extinction, the learning that happens is much weaker than the initially acquired fear, and it's prone to return due to various conditions. And one of those conditions is the lapse of time. So if we let some time pass and we present again these neutral stimuli, what we see is that some participants exhibit again a conditioned response to the blue light, even though they've previously extinguished that. And some participants manage to recall what they have learned during the fear extinction phase. Other manipulations that result in the return of fear is the presentation of these stimuli in a different context. So what you can see here is the presentation of the blue light, for instance, in the previously dangerous context, and this also can result in the renewal of fear, so the return of the conditioned response. And how we measure fear here is via physiological indices such as the skin conductance response, and also at the subjective level, like how much do these participants expect that an electric stimulation would follow after the presentation of the yellow or the blue lights. Now, if we look a little bit at the literature in animals in the gut-brain axis, we see that some scholars have administered prebiotics, which are a form of dietary fiber that gets fermented by the gut microbiota into short-chain fatty acids. And what you can see here is that these animals that consumed FOS, GOS, and a combination of both, which are prebiotics, exhibited a lower corticosterone response to administered stress. And another group has also found similar results when administering short-chain fatty acids directly into these animals. So you can see here the animals that have undergone stress in black and dark green. Those who ingested short-chain fatty acids exhibited a lower corticosterone response. And in relation to fear, this has also been explored, for instance, using the elevated plasmase, in which the animals exhibit or by default, they are afraid of the open arms of the elevated plasmase because it's more dangerous. But once they take prebiotics, they tend to spend more time in these open arms, and they therefore, or it's taken as an index of lower anxiety-like behavior. And we can, of course, see the opposite pattern as in spending less time in the closed arms. And then again, another study that has found that the systemic injection of butyrate, so one of these short-chain fatty acids, resulted in lower mean percentage of freezing, which occurred here during the extinction phase. So these are just a few studies, but they do give an indication that the consumption of prebiotics and its fermentation into short-chain fatty acids is capable of influencing stress and fear responding in animals. So what I wanted to do was basically to try to understand whether the relationship between the dietary fiber component of the diet and our mood and emotional responsiveness is mediated by the fermentation of of the dietary fiber into short-chain fatty acids by the gut microbiota. And if that was the case, then we would be expecting a causal relationship between short-chain fatty acids and stress and fear responding. So to mechanistically study the influence of short-chain fatty acids on psychobiological processes, we wanted to directly administer them into the colon, but to do so in an exact and in a prolonged manner is actually quite challenging. So what we have done is basically fill these short-chain fatty acids in a specific amount and ratio into capsules, and we then coated them in such a way would bypass the entire gastrointestinal tract until they reach the terminal ileum and then release their content into the colon, where they are normally produced as a result of the fermentation of dietary fiber. Of course, we have checked the performance of these capsules prior to running the study, and as a placebo, we utilized an inert dietary fiber. So for this study, I recruited 66 healthy male participants and divided them into 3 groups. They first came to the lab and underwent the acute stress and the fear conditioning and extinction paradigms, and then they were assigned to either consume placebo, a low dose of short-chain fatty acids, and a high dose of short-chain fatty acids daily for 7 days, and then they came one more time to the lab to perform these tasks again. So onto the results. First, we looked at the levels of short-chain fatty acids in the blood samples that we have collected. And as you can see here, there was a significant increase in the low dose and the high dose group in terms of the levels of acetate and propionate and butyrate. And especially for acetate and butyrate, this was actually in a dose-dependent manner. So this tells us that our intervention specifically the contents in the capsules were released in a dose-dependent manner, which is already a very important indication. Next, we looked at the cortisol response to the acute stress paradigm. And there what we were able to see is that participants in the low and in the high dose group exhibited a significantly greater decrease in the cortisol stress response in relation or in comparison with the placebo group. So that was quite intriguing to see. And then we wondered whether there is any covariation between the decrease in the level of cortisol here and also the increase in the level of short-chain fatty acids in the blood. So to try sort of to indirectly get an idea on whether the presence of short-chain fatty acids in the blood is important to the observed effect. And that is actually indeed what we found. So what you see here on the y-axis is the decrease in cortisol response, and these are all three groups grouped together basically. And the decrease in cortisol tends to be higher for those participants that had the highest increase in short-chain fatty acid levels. And that was the case for acetate, propionate, butyrate, and also total short-chain fatty acids. We also looked at the subjective stress responses. These are not shown on the slide, but there we did not see any effects as a result of the intervention. So we could conclude that short-chain fatty acid attenuated the cortisol, but not subjective responses to acute stress, and that the amount that was retrieved in the blood may be an important factor here. Now, in relation to the fear task, what you can see here is on the x-axis, the various phases that the participants performed during the fear conditioning and extinction task. And on the y-axis is the differential skin conductance responses. So this is the conditioned response. And at the phase level, we could see across all participants that the conditioned response was highest during the acquisition or the conditioning of fear, and then it went down during the extinction phase. There was also an increase in the conditioned response during the recall, indicative of spontaneous recovery, and also renewal of fear as expected. However, this was not modulated by the intervention, so there are no differences here between the groups. When we look at the subjective level, there's a slightly cleaner picture, of course, and you can see here that the expectancy ratings of the aversive outcome, so the mild electrical stimulation, was much higher for the dangerous stimulus, the blue light, if you remember, that tends to go down throughout the extinction phase and then return again during the test phases. But here also we were not able to detect any differences as a function of the intervention. So this suggested that short-chain fatty acids did not affect fear acquisition nor extinction at the autonomic nor subjective levels. So in conclusion, what we could find was that colon-delivered short-chain fatty acids attenuated the cortisol response to stress, but did not modulate the acquisition or extinction of fear. And this suggests that short-chain fatty acids may play a mediating role in psychobiological effects of microbiota modulating interventions, such as fermentable dietary fibers or prebiotics. And it seems like the magnitude of the increase in short-chain fatty acid levels in the blood may be a decisive factor in observing on effects on brain function. So this brings me to the end of my presentation. I hope I was not too fast, and I look forward to your questions.
Thank you, Boushra. It was a really nice presentation.
Thank you.
There are no questions for the audience whatsoever, but I might have a question about your last study. So let's say, short-chain fatty acids, is there any relationship to disorders like where there's a difference there in this take-up? For instance, Parkinson or other disorders?
Yeah. Yes, so what is actually more frequently done is looking at Alzheimer's and Parkinson's, autism and ADHD models in animals.
Mm-hmm.
And there you tend to see at the moment really depending on the model that is used, short-chain fatty acids can have different effects. So they could be positive or negative. But in humans, we have much less data. And mostly what happens is that the fecal samples of patients and controls are analyzed for levels of short-chain fatty acids and correlated with symptom severity, for instance. But there you also tend to see a lot of inconclusive findings, we can say. And this is why what we are doing right now is basically what we have done is administer short-chain fatty acids in physiological amounts, because what happens in animal studies, they tend to be administered in super physiological amounts. And so not following what would happen when dietary fiber, for instance, is consumed. This is what we tried to do here in humans, and I think there are a lot of still questions to be answered, both from the human perspective and the animal perspective, in relation to doses, in relation to the roles of each separate short-chain fatty acid as well.
Indeed. I guess that's now work of your new postdoc, right? That you started?
That will be in humans, yes.
Yeah, okay. A small question from in the chat. It says, I think your sample included only male participants, right? Would you expect similar results for women, or is there a difference in metabolism or take-up of?
The reason why we included males is because we wanted our primary outcome to be the cortisol response to stress, which tends to be quite variable in females. And we also wanted to do a 1-week intervention. So there you would not be able to control for menstrual cycle phase. And also, if you were to recruit women on contraceptive pills, for instance, you already have a very blunted cortisol response to begin with, so you're less likely to see a change as a function of the intervention. Whether I would expect a similar change, I think that is something that is an experimental question. I definitely hope so, especially from the fact that depressive and anxiety disorders tend to be more common in females. So this is something definitely on the agenda. But here we had to go with as homogeneous sample as possible, and I can tell you it's not homogeneous, but it is more homogeneous than including more both sexes. So
yeah, I think you should, because
starting point.
50% of the global population are women. So
of course, yeah, I think it was like when I started this research, I was it was quite risky, of course, and then when I was looking at previous studies in humans who administered like dietary fiber or probiotics, they were also primarily in males and also in male animals. So it was a bit difficult to like make hypotheses about females. And yeah, unfortunately, I had to commit the same mistake. But
you have to break the cycle.
I have to break the cycle. Yeah, for sure.
Okay, I think we should continue this discussion in the networking session, for instance.
Of course.
And other people also. Are you invited for a short break now of 10 minutes? And then you can go to your poster session, D, E, F, and G. So hope to see you there at the posters. And then when you're finished, you can come back to this stage again, because then Mathieu will do the closing. Thank you, and see you later.
Thank you so much.
Bye.